Have a pleasant day to all of you, especially to our Professor Loreline Joy Tornos. Today, I'm going to present to you the gas exchange and movement in the post-harvest. Respiration is the oxidative breakdown of complex substrate molecules normally present in plant cells, such as starch, sugars, and organic acids, to simpler molecules such as carbon dioxide and water. Concomitant with this catabolic reaction is the production of energy and intermediate molecules that are required to sustain the myriad of anabolic reactions essential for the maintenance of cellular organization and membrane integrity of living cells. Respiration plays a major role in the post-harvest life of fresh vegetables for the following reasons. First, loss in substrate. Second, oxygen requirements. Third, carbon dioxide production. And fourth, release of heat energy. Gas exchange between a plant organ and its environment follows Fick's first law of diffusion. The sequential steps are diffusion in the gas phase through the thermal system, such as cuticle, epidermis, stomata, lenticels, etc. Diffusion in the gas phase through intercellular spaces. Exchange of gases between the intercellular atmosphere and the cellular solution, like cell sap, or vice versa. And diffusion in solution within the cell to centers of oxide consumption and from centers of carbon dioxide production. This exchange is a function of the resistance of the thermal system to gas diffusion, the distribution of the intercellular spaces, the tortuousness of the diffusive path, the surface area across which diffusion can take place, the solute concentration of the tissue, and the gradient in gas concentration established by the respiratory activity of the tissue. Carbon dioxide produced within each cell will raise the local concentration, and the gradient produced will drive diffusion of carbon dioxide outward toward the lower concentration near the cell wall surface adjacent to the intercellular space. Diffusion of carbon dioxide into the intercellular space continues toward regions of lower concentration until it reaches the intercellular space below the thermal system. From there, carbon dioxide moves through the cuticle or openings in the commodity surface to the ambient air. Gradients of oxide within plant tissues are established in a reverse but analogous process to that mentioned for carbon dioxide. In senescent tissues, oxide diffusion may become so impaired if the intercellular spaces become filled with cellular solution that anaerobic conditions develop within the tissue. The rate of gas movement depends on the properties of the gas molecule, the magnitude of the gradient, and physical properties of the intervening barriers such as thickness, surface area, density, and molecular structure. Both the solubility and diffusivity of each gas are important for its diffusion across barriers. Carbon dioxide moves more readily than oxide, while diffusion rates of hydrocarbon and carbon dioxide are similar. In leaves, gas diffusion is regulated by control of the stomatal aperture by guard cells But most bulky organs have no functional stomata or other active controls of gas exchange. The number of other factors influence gas diffusion in bulky organs. They have a much lower surface to volume ratio than leaves. The distance over which gases must diffuse in the tissue is relatively large compared to leaves, and respiration, not photosynthesis, is a major metabolic process. Example reactions producing carbon dioxide and consuming oxide rather than the reverse. The internal concentrations of oxide and carbon dioxide in plant organs depend upon the maturity stage at harvest, the current organ temperature, the composition of the external atmosphere, and any added barriers. Maturity stage influences the respiration rate and the components of the thermal system that affect gas diffusion, such as the development, composition, and thickness of the cuticle, epidermal hairs, trichomes, and lenticels. Increased temperatures raise the rate of respiration, and in response, the internal carbon dioxide concentration increases as the oxide concentration decreases. If all other factors are held constant, and if the gradient in gas concentrations is the driving force for diffusion, then the concentrations of oxide and carbon dioxide within the tissue will fluctuate in accord with fluctuation in the external atmosphere. For example, a change in the external concentration from 21% to 15% for oxide, a decrease of 6%, and from 0.03% to 3% for carbon dioxide, an increase of 3%, would cause a concomitant decrease in internal oxide by 6% and an increase in carbon dioxide by 3%. However, these changes could affect respiration and produce different outcomes, especially if the gas concentrations exceed the tolerance limits. So there are three types of barriers to gas exchange that affect the post-harvest handling of fresh produce, as we can see in the figure 1. At the level of the commodity, the structure of the thermal system, such as thickness of the cuticle, wax composition and arrangement on the surface, number and distribution of stomata, lenticels, and bricks in the epidermis, represent the first significant barrier to gas diffusion. Resistance to gas diffusion can be increased by added barriers such as wax coatings and wrapping with polymeric films. The package in which the commodity is shipped can be additional barrier to gas diffusion. Its significance will depend upon the permeability of the package materials, extent of ventilation openings, and use of plastic liners within the package. Furthermore, the degree of gas tightness of the transit vehicle or storage room will also affect gas exchange with outside air. All these barriers must be considered from the standpoint of providing the optimum oxide and carbon dioxide concentrations within each commodity that will maximize its post-harvest life. Fick's first law of diffusion states that the movement or flux of a gas in or out of a plant tissue depends on the concentration drop across the barrier involved, the surface area of the barrier, and the resistance of the barrier to diffusion. A simplified version of Fick's law can be written as what you can see in our slide. The resistance of tissues and organs to diffusion of carbon dioxide, oxide, and hydrocarbon has been investigated using the steady-state approach. The production or consumption rate of the gas by the organ and concentrations of the gas in the internal and external atmospheres are determined. Then, the resistance is calculated as follows. Resistance equals concentration gradient over production or consumption rate. So the accurate measurement of the internal concentration of gases is often difficult. Although internal samples are easily withdrawn from fruits with internal cavities, such as cantaloupe, melons, reticulatus group, extraction methods used to determine the internal atmospheric composition of some bulky organs, such as potatoes, are less satisfactory and can yield inconsistent results. So there are researches on gas exchange. Reducing gas exchange of fruits with surface coatings. In the abstract, surface coatings can increase a fruit's skin resistance to gas diffusion, modify its internal atmosphere composition, and depress its respiration rate. Effects on transpiration tend to be comparatively small. A steady-state mathematical model which considered diffusion through cuticle and pores separately was used to investigate two possible mechanisms by which coatings achieve these effects, either by acting as a film wrap or by blocking pores. It was concluded that coatings mainly exert their effects on skin resistance to diffusion of the permanent gases by blocking a greater or lesser proportion of the pores on the fruit's surface. Both covering of the cuticle and blocking pores were important for resistance to water vapor diffusion. Coupled with a proposed differential resistance of the skin to oxygen, carbon dioxide, and water vapor, this accounts for the observed effects of coatings on internal atmosphere modification, respiration, and transpiration rates. Coating treatments which achieved substantial reductions in transpiration rates were associated with the fruit becoming anaerobic. Inherent variability in skin resistance to gas diffusion and fruit respiration rate, and the varying proportions for pores blocked by coating appear to be responsible for the highly variable response of individual fruit to a given coating treatment. Surface coatings may therefore be better suited to treating fruits destined for processing, in which fruit-to-fruit variability may be less critical than to fruits destined for the fresh market, in which uniformity of individual fruit quality is of paramount importance. Another research study titled Gas Exchange, Ripening Behavior, and Post-Harvest Quality of Coated Pears that you can also search on the Google. For the conclusion, an adequate oxide concentration must be available to maintain aerobic respiration. This should be considered in selecting the various post-harvest handling procedures such as waxing and other surface coatings, film wrapping, and packaging. Accumulation of carbon dioxide produced by the commodity in its ambient atmosphere can be beneficial or harmful, depending upon each commodity's tolerance to elevated carbon dioxide levels. For some vegetables, increasing the carbon dioxide concentration around them in a controlled or modified atmosphere can be used to delay senescence and retard fungal growth. And these are my references. Thank you.